Hi guys and welcome to Learn Extra Live. It is a Wednesday and it is grade 10 life science. I've got Knox next to me, one of the top 10 presenters, and we are so excited he's in the studio. How are you, dude?
I'm finding yourself, Indy.
It's so good. I'm so excited you're joining us today.
I'm so excited too. Actually, I missed all the mindsetters at home today.
Oh, I'm so glad. And so
Yeah.
now
I just
you
missed
actually get
the
to be in an
interactions.
entire show.
Yeah, that's
Yeah.
really cool actually.
Yeah. So now, guys, I think maybe you should tell them where to find us on Facebook.
Okay, remember guys that if you have any sort of question, please don't hesitate to post it on our Facebook page. That is www.facebook.com/learnextra. That's www.facebook.com/learnextra.
That's exactly where it's at.
Yeah.
And also they can tweet us at Learn Extra. Drop your questions there. Eugen is on the show today and he will be answering all of your questions and taking you through, I think it's cell division and mitosis. How are you, Eugen?
I'm great, and yourself, Indy?
Oh, so glad that we are here because I think life science is one of my favorite subjects and I know that
Same
it's one
here.
of yours.
Yeah.
Tell us a little bit about yourself, Knox.
Okay, my name is Doza Monani. I'm 18 years old, turning 19 this year. I'm from Soweto and I study at Wits. I do Bachelor of Arts in Law.
And you loved life science, right?
I love life sciences so much. I did life sciences in my matric year.
And why is it that you love life science so much, do you think?
I think it's very, very real, you know, it's very tangible. But over and above that, you just get to learn things that you don't know. You just go deeper into biology and life, basically.
So, Eugen,
Awesome.
you need to ask Knox lots and lots of questions. And mindset says, do you know what? Actually, please post your questions on the page and Knox will be on the page answering all of your life science questions. Let's put this buff to the
That's
test.
not fair.
And it is totally fair. It is totally fair. I cannot wait. Okay, so Eugen, let's get going. Let's cell division, mitosis. I think we're ready.
Awesome, guys. So thanks for that, guys. And remember, we're coming to the grade 10 session. We're doing life sciences today. We're looking at mitosis. And very quickly, to chat about what we've done over the past couple of weeks. So we started with organic compounds. We went on to looking at cells, different types of cells. We looked at plant cells. We looked at animal cells. Now we're going to look at cell division. And then next week, we're going to look at how cells make up tissues. And it's important for us to be able to see how this was scaffolded so that we understand how one concept leads to the next. And that's essentially what life sciences is. It's about understanding life and exactly what Knox said. Understanding life and being able to relate to what you study in society itself. So why do we study cell division? We're constantly growing, we're constantly having injuries that are repaired, and hence we need to understand what happens in our body. And we refer to that as cell division. And there are two types of cell division that we look at. One is mitosis, and the other is meiosis, which is done in grade 12. But I'm going to do exclusively today lots of mitosis. I'm going to do the stages. I'm going to explain that to you. I anticipate that you guys are going to have lots of questions. If you haven't done this section at school yet, it's good. Engage with us, guys. It's going to help you understand when you get to the concepts at school. So let's get to what cell division really is. Okay, so let's look at what we have in store today. So we're going to look at the related terminology, and as I suggested, and I always make this point very clear, is that create a list of terminology. I know that all the guys that I teach at Saturday school, guys, I mentioned this, I know you guys are watching, and it's for you. I've created this list of terminology. Make sure that you have this list. Write down the important words that you need to so that you can easily refer to that when you're studying. We're going to discuss the cell cycle today. We're going to look at the purpose of cell division, and that's very important to understand why cell division takes place. To consider what mitosis is as a type of cell division, we're going to look at the various stages of cell division, and we're going to consider what happens when cell division goes wrong. And cancer is something that affects us all. To understand why and how cells divide and cell division goes wrong, and the consequences of such, and the treatment of cancer. And we're going to do lots of revision questions, and that they are out there for you guys. You're going to post many questions to us. We're going to try and answer them, as well as I've got questions for you which you can refer to. So let's look at the terminology list. So we're going to look at terms like mitosis, centromere, anaphase, karyokinesis, cell cycle, interphase. Again, we're going to look at telophase. What is the term malignant? We're going to look at chromosomes, prophase, the word benign, tumors, chromatids, metaphase, cytokinesis, and cancer. So these are just some of the terms. I know that they might be unfamiliar to you at this stage, but as we go through, I'm going to explain these terms in detail. But the fact that I've got them down, guys, if you download these notes, you have these, you can write a simple definition for each of these terms as you go through the section. And that's essentially why we have these keywords that we have. so that as we go through, we can elaborate and define and describe each of these terms which are important in this section.
Again, while you're on that note, I just wanted to tell the mindsetters that all these notes are available on our website, and it is learnextra.co.za/live. You can get the notes we're going over right now. You can get all of last week's videos. You can get our schedules. So that is the most important link to know, guys. I've actually just posted on the page, so get it there. I think, Eugen, sorry for interrupting. Carry on, but I just had to tell them.
No, it was important that you guys know that it's there, guys, so you can simultaneously look at these notes while I do the explanations live for you. So let's look at what cell cycle is. So organisms have a life cycle. Organisms are made up of cells. So if an organism has a life cycle, there's a process of it going through cell division, and each cell will have a cycle or a life cycle as such. So to understand how cells divide, they go through a series of stages of development and specialization, and that's essentially the different stages of development and specialization is referred to as a cycle, and often a cycle refers to a series of events that start at a specific point and that brings us back to that point again. So the cycle starts when the cell forms and ends when a mature cell divides into two daughter cells. So refer to the mature cell dividing into two daughter cells. What is said is that life starts from one form, and that form divides. So after fertilization of an egg and a sperm, we have a zygote, and from that point in time, the cell divides constantly and divides and specializes to become a complex multicellular organism, or an organism or a human as such. So when we look at complex organisms, remember that they all started with a one cell stage, and that cell constantly underwent cell division and its cell cycle to become specialized. And essentially we're going to look at a phase in that, which is mitosis. The cell cycle has three parts, and it's important for us to remember these three parts. It goes through a phase called interphase, which is cell growth, and that is when the cell grows and specializes and becomes specialized for its specific function. It also has a second stage, which is mitosis, and this is the stage when the cell needs to divide and form another cell, so it splits up into two cells, and essentially that's the third stage of cell division, or the second stage of cell division. And the third is cytokinesis. Now the term cytokinesis is made up of two words. If we break this word up, cyto, we can look at it's got cyto, and that comes from the word cytoplasm, which we've done already, and kinesis, guys, refers to, I'm just going to change, kinesis refers to the chromatin network. So the movement. So cytokinesis refers to the movement or the splitting up of the cytoplasmic material, which happens at the end of cell division. So essentially there are three phases that the cell undergoes through its life cycle. That's interphase, mitosis, and cytokinesis. Let's move on to the details of cell cycle, and we're going to look at interphase and chromosomes. Now, before we start, we often refer to the word as the term chromosomes, and often we don't understand what chromosomes are, guys. But I'm going to throw maybe Indy and Knox into the deep end and ask them what chromosomes are and how many we have.
So I think you should throw Knox into the deep end. How many chromosomes do we have?
Isn't it 46?
Awesome. And that is a good way of pushing it, brushing it off, Indy.
Just pass it on over.
Very tactful.
No.
But remember, guys, we've got chromosomes in our cells, and we have 46 chromosomes. So when we look at a cell, and if you remember from the last 2 weeks, we looked at the nucleus, and we saw that in the nucleus there was a whole lot of chromatin network, and that chromatin network is basically DNA. How does that chromatin network form chromosome? Is a process that happens during interphase. So when the cell prepares to divide, and often use this to explain the learners that I teach, and I tell them that if I give you a bowl of spaghetti, which is obviously got lots of strings and strands, and ask you to divide that equally, what is the best way to divide it equally so that the two people that you divide it to get equal and exact amounts? What would you do, Indy?
Individually, take a fork and a knife and just split them down the middle.
Yeah, absolutely, and that's exactly what happens. But it wouldn't be exactly accurate, and unfortunately,
Knox knows.
I'm going to save Knox on this one here. Okay, but what happens is that remember that the chromatin network is a ball. It's a whole lot of DNA that's wrapped up and entangled, and for the cell to be able to randomly divide into two halves, it cannot do that. And hence, what it does is that it's quite intelligent. It wraps these chromatin networks up into little strands, and it starts doing that. And eventually to become compact so that it forms a structure known as a chromosome. So in anticipation of cell division, it starts coiling the DNA around histone proteins to become a compact structure, which is essentially a chromosome, which is wrapped around histone protein. So here, if we look at this diagram, we've got a chromosome showing we're showing the parts of a chromosome. Here we're seeing a little ball called the centromere, which is a protein that holds the two strands together. Here we've got one strand, which is referred to as a chromatid, and here we have the other strand, which is referred to as a daughter chromatid as well, and this collectively is referred to as a chromosome. So we have 46 of these in our body, in each of our cells, except for the sperm and egg cells. We'll discuss that in detail later, but essentially the nucleus in interphase will prepare for cell division by arranging the chromatin network into compact chromosomes, and in that process it needs to undergo replication. And replication basically means the copying of the genetic information into another copy, so that when we divide into two cells, each of the cells have an exact copy of the original genetic information. So at the beginning of interphase, the cell grows quickly, more organelles are made, and there's an increase in the number of chemical reactions, and this is because the cell is undergoing its normal physiological processes. It may become specialized for a specific function that the body may need, and hence we see cells becoming specialized for different functions. Towards the end of interphase, guys, just at the end, as I mentioned, the chromatin network makes a copy of itself, and that process is called replication. And for you guys in grade 12 that are watching this, replication is a process of DNA forming a copy of itself. And remember that we don't do this in grade 10, but in grade 12 we do replication. And you got to remember this for grade 10s. As you get into grade 12, we've done replication here, we've mentioned the concept, but we do the explanation of the process of how DNA copies itself, and that is replication when we get to grade 12. So the DNA replication takes place so that the chromatin network copies itself. The chromatin network then coils up, as I mentioned, to form a short chromosome. So it forms a compact chromosome, and it wraps itself around proteins called histone proteins. There are chromosomes in the nucleus of every cell. Guys, in the body, every cell has a chromosome. Indy, how about let's challenge our learners to tell us which one of the cells in our body does not have any DNA. So that's a challenge question out, and let's see if somebody can give us the answer.
I love that. I'm going to put it straight onto the Facebook page, and then let's make that a challenge question.
Sure.
Okay.
Hey, I like that.
So which cell in the body does not have a nucleus? Okay, and I'll give you the answer as we go through towards the end. And I know you guys are bright. I'm going to get lots of answers, but let's see how many correct answers we do get. Okay. So what happens at the end of interphase? We have two identical strands called chromatids, and these are the chromatids that I've highlighted here that are joined together at one point called the centromere. So sort of, if you were to put your elbows together, guys, and you were to stick them together, this was an exact copy of that arm, and we refer to these as daughter chromatids held together by a centromere. And that's essentially what happens during interphase, where we have the centromere forming. Okay, we're going to move on to mitosis and the purpose. We're going to do the details of this, but why is mitosis important? We know that we grow every day. We fall, we get injured, and hence we need to repair. So it's important for growth and development of organisms, as we all have started from a single cell, and as we grew, we divided, we became specialized, and that is essentially what happens. So it's responsible for growth, it's responsible for repair. So whenever you're injured, whenever tissues and organs are injured, repair of organs and tissues happens by a type of division we know as mitosis. Okay, it's also important for reproduction, and this type of reproduction is asexual reproduction, guys, and this happens in organisms such as bacteria and protists, and this is by a process called binary fission or budding, where a cell splits up to form two cells, and we have this at the end. So it undergoes a series of stages where it splits up and buds off to form two cells, and that is a kind of cell division where we have from one cell two new organisms being formed. Okay, where does the process of mitosis take place? And this takes us back to the cell structure. And I know that we've done this last week. We said that the nucleus contains the chromatin network and the DNA. So essentially, when we divide cells, we need to ensure that when we have two cells, the nucleus that has divided is an exact copy of itself. So essentially, it happens in plant cells. Mitosis occurs in the apical part. So when we prune our trees at home, we cut the tips off, and the tips are the parts that actually grow in height. We also have branches that grow laterally. So at the tips of these branches are meristematic tissue, or tissue that constantly divides by mitosis that actually causes the plant to grow. And I know you guys would have noticed that sometimes gardeners are able to manicure their trees to grow into a specific shape, and that's by using the ability to trim the tips of the meristematic tissue on the branches as well as on the side lateral branches, so that the plant can be directed into a specific shape or size. In animals, guys, sorry about that, it happens in specific places like in the bone marrow, the skin cells. Some tissue constantly is replaced by mitosis. You know that skin, guys, now that winter is approaching, you know that your skin becomes dry, and that obviously exfoliates, it shreds off, and that process of exfoliating is old cells being replaced by new cells. And I know, Indy, I'm going to bring in a bit of beauty therapy in here. My wife's a beauty therapist, and she talks about exfoliation.
Oh yes, exfoliation is key, at least once a week, ladies.
And will you be able to tell us what exfoliation is?
Exfoliation, it's when you're washing the face, but you're washing it with something kind of slightly rough, particly, that actually takes off the top layer, the dead skin cells on your face.
So, yes, awesome. So, and that is essentially understanding what life sciences is, guys. Being able to understand what we do in everyday life. So, the process of exfoliation, I know my wife does it regularly.
Do you, Eugen?
Yeah.
You
no,
do
we
exfoliate. I can see by your
reserve that.
complexion. You've got a beautiful complexion.
Oh, no. Yeah, it's the light. It's the effect of the light. Yeah, so it's removing the dry skin from the surface of the body, and that's being replaced constantly by the epidermal layer that generates new skin. And that process is called, is caused by mitosis. Other organs like the liver and the skin cells, these constantly divide as well and repair. Guys, we've done the introduction to this topic. We're going to go for a break. I'm going to hand you over to Indy.
Fantastic. I think during the break, Knox and I were going to have a little bit of an exfoliating session. And when we get back, we'll be all bright and sparkly and looking lovely. Hey?
Yeah. So
Okay.
guys, make sure that you join us after this.
Hi guys, and welcome back to Learn Extra Live Life Science. If you have been missing out, where have you been? And at least they're joining in now, right?
They are, and I have to say, you guys actually missed out. But remember that you guys can still sort of catch up by downloading all the notes from our Mindset Learn website.
And that is learnextra.co.za/live. Learn that link, guys. It's where you get all of your schedules, it's where you get all of your show notes, and it's where you're going to get all of your videos. If you click on the links, we've got a YouTube channel where all of our Learn Extra Live videos get put on there, so you can go play, pause, share it with your friends, use the resources.
But Indy, I haven't had any sort of questions on Facebook from the Mindsetters.
What is
so
this
guys,
about?
please remember, guys, post all those questions. We can't wait to hear from you. We just want to answer you and interact with you. Remember, that's www.facebook.com/learnextra.
Perfect. Awesome. And also chat to us on Twitter @LearnExtra. Knox is a fundy, guys. He's a life science fundy. And I mean, obviously Eugen is as well. But you can send us through questions. Use us. You have the best teachers here. Use them. Ask questions. The only silly question is the one asked after exams. I sound like a broken record. It's the truth. It's the truth. Okay. So Eugen, what is mitosis? Is the question.
So let me just fill in the viewers in terms of what happened during the break. So we had two presenters that went and had an exfoliation. So what do we have now, guys? We have two bright sparks. Right.
Beautiful.
So just in terms of, and probably we're not getting many questions, so it's because maybe the grade 10s have not gone to this section yet. But guys, it's still, if you're paying attention and if you're listening to this, remember the advantage of listening. I know some schools have started with cell division. If you haven't done this section, guys, here's a chance to listen. Ask us those questions. Maybe you can go and ask your teachers as well, or give them the questions that you're asking us in terms of after we answer them to you. It doesn't have to be cell division as well. It could be anything that you think is related to cell division. Let us have those questions. You've got to ask questions, guys. And that's the only way to learn. Okay, so let's look at what is mitosis. So mitosis is linked to cell growth, and so we're going to go back to that. Okay, so mitosis is linked to Cell growth, it is a process of cell division where a mature cell divides into two new identical cells. Mitosis usually takes an hour to two. And it is a continuous process that never stops, and a cell will constantly divide through its life cycle, and it will divide to form a new cell. It will further divide to form further new cells, and hence, if you were to describe what mitosis is, this would be a very good definition or description for explaining the process of what it is. Okay, so if we look at the next slide, the stages of mitosis in plant and animal cells. So there are two types of divisions that are important that we look at, and we refer to that as karyokinesis. I mentioned this term earlier on, but we hadn't defined what it is, and cytokinesis. Now the word karyo refers to carry on, or refers to the nuclear material. So when we talk of karyokinesis, we refer to the division. Kinesis is the movement of the nuclear material, or the division of the nuclear material. So it's a division of the nucleus. So it's division of the chromosomes moving up into two different cells, and we have it followed by cytokinesis. Cytokinesis refers to the division of the cytoplasmic material, and that is when the cytoplasm follows nuclear division, splitting up into two cells. Okay. To make it easier to describe cell division, we divide cell division into 4 phases. So we call them phases, and here we've got them prophase, metaphase, Anaphase, and Telophase or Telophase. And guys, it's often difficult to remember. There's so much in life sciences that you've got to remember, but it's very easy to use the acronym PMAT. Okay, so PMAT, and that's essentially how you remember this. So PMAT would be the order in which you remember it. P meaning prophase, M meaning metaphase, A anaphase, and T for telophase. And there's an even easier way of remembering what each of them. M for meet, A for at, and move apart as well. So I'm giving you some keywords to remember, and T for telo, which refers to the poles. So there's movement apart towards the poles, and that's essentially what the stages describe. Pro would be the first. Okay, so very, very quickly, if you're confused by the complex terms, pro meaning first, meta would mean meeting, ana would mean moving apart, and telo would be to the and similar to, remember, telephone poles, moving towards the poles of the cell. So we're going to look at each of these stages, and when we do these stages, guys, it's important to remember that we should be able to identify the characteristics of a stage, and that's essentially what you're going to get. Your teacher will not ask you to explain the stages. He will give you a diagram, or she will give you a diagram and ask you to identify which stage it is and say why it is that stage. And essentially that is what I'm going to try and teach you today. I want to try and teach you the key characteristics of each stage, so that by looking at a cell or even an electromicrograph or a photograph of cells in division, you would be able to make a call as to which stage it is. And that's important in understanding the process. So very quickly, let's look at the cell. So here we see a cell, and here we see the DNA material that has duplicated or replicated to form a copy of itself, and that's essential. Before cell divide, during interphase, and that's the phase we're looking at, it will replicate, it will copy the DNA, so that when it gets into the next stages, it starts dividing the DNA, so that the cells that form are identical to the original cell. Okay, so here we're seeing cells that have the chromatin network now forming chromosomes. So the cells may appear inactive during this stage. They are quite the opposite. That means they become very slow in terms of their movement. This is the longest period of the complete cell cycle, in which the DNA replicates and the centrioles divide and proteins are actively produced. So during this stage, guys, the cell is actually now preparing for cell division, forming more copies of the DNA. It starts to slow down the cell cycle. This is probably the longest stage that it spends its life cycle in. Let's get on to the actual stages that I discussed earlier on. So what is prophase? Pro, I said first. So let's look at the first phase. In prophase, here you can clearly see chromosomes are made, and each chromosome has now 2 chromatids. So we can clearly see here we're seeing, it might not be very clear, here we're seeing a little chromosome, here we're seeing, I'm going to take that off, guys, so that my pen is too, but here you can clearly see what I've drawn here, guys. Oh, let me do that again. taken that and I've drawn you here. Here you can see a centromere, and you see 2 daughter chromatids that are attached to the centromere. And essentially, when we look at a cell, this has 1, 2, 3, 4 chromosomes that have formed chromatids. And the key way to identifying the number of chromosomes in a cell would be to count the number of centromeres. So if you count the number of centromeres, you'll be able to determine the number of chromosomes. Okay, so let's look at a cell that is showing the formation. Here we see, not very clear, but here we see the formation of chromosomes in a cell. So in prophase, the centrosome is made up of 2 separate centrioles. Fibers form. Fibers, we call them spindle fibers, form between the centrosomes. So the centrosomes are, if we look at this, guys, here we're seeing these centrosomes, or we call them sort of satellite bodies that move towards the opposite poles of the cell. So these centrosomes start moving towards the opposite poles, and they become sort of like the north and south poles of a cell, essentially on either end. And from them, we have sort of, I'm going to try and draw this right here for you. We're going to have the centrioles moving to the opposite poles, and from them, spindle fibers developing through from one pole to the opposite pole. Okay, so centrosomes move to the opposite poles. Here we see them moving to the opposite poles. Each chromosome is visible as 2 chromatids joined by a centromere. And that's essentially what we can see in this diagram. We're seeing each chromosome being attached by a centromere holding 2 chromatids together. Okay, so let's move on to the next stage. And if you remember mat, M-A-T, and metaphase would essentially mean meeting. So there's something happening here, there's a meeting happening, and obviously the meeting of the chromosomes. So when we look at this diagram here, guys, now we see that at the poles, which I mentioned earlier on, we have your centromeres at the poles, and from them, your centrosomes at the poles, and from them we have these spindle fibers that are growing towards the opposite poles. And along the equator, guys, I'm going to take this off. You can see the arrangement of the chromosome. I'm going to do this again. Here you can see the chromatids in the middle, along the equator in a single line arranged. So essentially, we here in this cell, we're seeing that there's 1, 2, 3, 4 chromosomes arranged or meeting along, and we call this the equator of the cell. So you got to remember that it's similar to, imagine this was the Earth. You had a North Pole and the South Pole, and along the equator, you've got the chromosomes meeting along the equator. So these are chromosomes along the equator. And why would we want to meet along the equator? Indy? Knox? Why would we want to meet at a point?
I think that's because they need to separate in the middle.
Absolutely. Awesome. So they need to be able to move to the opposite poles from a central position, and that is when we actually see now the cells splitting up and the chromatin starting the chromatin or the chromosomes moving to the opposite poles. Okay, so I'm going to show you a photomicrograph of
And
this.
again, I think Knox has a question.
Yeah, we talked about the centrosome, right? Do they only form during the process of mitosis, or they're just always there in the cell?
Okay, good question. So the question is, guys, you heard the question, do these centrosomes always form? They actually form only during cell division. So they are in the cell. If we go back to, I'm going to go back a few slides to show you exactly. Here we're seeing these satellite bodies. So the centrioles normally lie very close to the nucleus, and it is these centrioles that start migrating. So there are 2 tubes that lie at right angles to each other, and during cell division, in preparation for prophase, they start moving to the opposite poles as satellite bodies. So they lie very close to the nucleus, and towards cell division, they start migrating towards the opposite poles, as I showed you in this here, where they start moving, as we see here, moving to the opposite poles. And essentially, the process of cell division and the formation of spindle fibers is controlled by the centrosomes. Okay, does that answer your question, Knox?
Thank you very much. It
Sure,
does.
very hard question that was for me at this time. Didn't anticipate.
Hey,
You see,
good, good, we're getting you back.
yeah, the exfoliation definitely helped. We've got some bright sparks now. Okay, so here we're looking at metaphase, guys. And look at if you look at this electromicrograph, guys, I'm not going to draw over the middle, but if you look right in the middle there, you can see that there's an arrangement. And here you can see, this is what the centrosomes or the satellite bodies, and from them we can see spindle fibers forming and running to. And in the middle of the spindle fibers, the spindle fibers are attached to the centromere. And that's a very nice diagram showing you the centrioles and the spindle fibers running through them. Okay, so when we look at the next phase, anaphase, guys, and remember anaphase, I said ana would refer to moving apart. Now we've arranged at metaphase, we've met along the equator. During anaphase, we're going to look at how they move apart. So during this stage, you can clearly see what happens. So we've got these chromosomes that we arranged. I'm going to very quickly show you how they arrange themselves along the equator. And this is what happens. Okay, so we've had them now pull apart and, sorry, let me just do this again for you. Take this off here. Okay, so I'm going to go back just to show you this, how this slide looked like. Here we see the arrangement along the equator, and here we see the chromosomes. Here we're seeing the chromosomes arranged like that, and through them we have the spindle fibers that run. So remembering that picture in mind, guys, we're going to look at anaphase, and this is essentially what happens in anaphase. We find that the centromere is split, and we find this movement to the opposite pole. And that's essentially what is happening here. So this splits up, and you find that the chromatids are pulled to the opposite poles. And essentially, a chromatid is a daughter chromatid would split up, and that will be a copy of the chromosome. And here we can clearly see along the equator, guys, that's the equator, we can see that the chromatids are pulled to the opposite poles. And it's very important to remember that each chromosome separates into its sister chromatids, because these are identical to each other. They come from the mother chromosome as such, to call it. And this is by the action of the spindle fibers. So imagine elastic rubber bands that are contracting, and these split up, and they pull the chromosomes, the daughter chromatids, we call them sister chromatids or daughter chromatids, to the opposite poles. And thus, at the end, we find that we have now the chromosomes split up into daughter chromosomes being pulled to the opposite poles. And that's why the opposite poles would mean that they're moving apart. So during anaphase, they've moved apart to the opposite poles. I think we're almost in for a break. Before we get on to telophase, guys, we're gonna go for a short break. I want you guys to rehydrate yourselves, come back fresh, post those questions, and let's get some answers.
That's exactly what we're going to do. There are a few guys posting on the Mindset Learn Facebook page. What I also tell them, Knox, is that this Facebook page isn't just for us, it's also for them.
For them, true.
It's a community of mindsets, as it really is, team mindsets. And if they see a question on the page that they understand, they can help each other out, because sometimes it's so much fun learning by helping other people.
I guess it's just one of those acts of kindness initiatives that we're trying to actually push.
Exactly. Maybe we should quickly talk about random acts of kindness.
Sure, please fill us in.
Yeah, tell us. Okay, well, you've already got the T-shirt, so you don't need to win another one, do you?
No, I'm fine.
So, guys, what it's all about, it's about passing on, forwarding the kindness, random acts of kindness. You can win this awesome hamper from Simba Chips and all these amazing Cheetos. They literally are so many, the packet's like this big.
It's a month's supply.
Yeah, literally, forever. You get this awesome Cheetos little notebook, lanyards from Mindset Learn, this awesome T-shirt, and 5 CDs during the month of love this February. And all you need to do is go and find on our events page. It's Pay It Forward. Post your random acts of kindness. Sometimes it can be really small. It's about helping each other out. If you see somebody that's struggling on the road carrying a heavy bag, help them. Asking somebody how their day is even a random act of kindness.
Yeah.
Just post your pictures or your videos or even what your random act of kindness has been on the page, and you could win this awesome hamper. We are giving away this hamper today in one of our shows, so make sure that you keep your eyes peeled, especially if you have entered and posted your random acts of kindness on the page. We'll see you now after the break. Hi guys, and welcome back. I hope that you are tuning in grade 10s. I think you again said that they're all being very, because I think shame. Knox is saying, but no one's talking on the
Yeah,
page.
it's really sad.
But then we were talking about that a lot of you might not have even started this work yet. So we're forgiving you for not answering or asking lots of questions, right?
But again, remember that you guys still have to download all those notes, even if you have not done the topic with your teacher at school.
Yeah, definitely. What tips would you give the grade 10s now that you've got benefits of hindsight, now that you're out of school?
I think life sciences is a beautiful subject, but unfortunately at the same time, there's a lot of content to go over, you know? So a lot of the time you might want to work around what Eugen said earlier. Try to come up with abbreviations, you know, like PMAT and that sort of thing to help you guys remember everything. Because often they'll want you guys to describe steps, to go through all the stages. So it might be quite beneficial for you guys at home to make sure that you get abbreviations, acronyms, symbols, just to make sure that you put in all of that content into your brains.
I think those are very wise words, right,
Absolutely.
again?
Yeah, it comes from somebody who's just been through the process.
Yeah.
I mean, and he's just Knox is just out of school. Not just out of school, but he's in the process of constantly learning, and you got to be innovative. You've got to work smart rather than work hard. And that's essentially what it is. So try and make learning fun and make it easier. It's not boring, guys. So remember that. So we're here. Let's continue, guys. I hope we're going to get some questions. I know that we got lots of answers to the challenge question that I posed, and lots of you are correct. We're waiting for more to post some answers. Okay. So before we went into the break, we looked at Anaphase, and we said characteristics where that the chromosomes split up into chromatids or daughter chromatids, and they moved apart to the opposite poles, as we clearly see in this diagram. The next phase, or the last stage, would be telophase. And I asked the kids that I teach, or the learners that I teach, to remember the word telephone poles, and that's how telophase. So this refers to poles, and obviously from movement to the poles. So here we find that the cell now starts dividing to form the poles. And so we see two significant things happening here. We see the end of karyokinesis complemented or simultaneously fitted in with cytokinesis. So we're seeing now the division of the nucleus, the last stage, telophase, and we're seeing the division of the cytoplasm, sort of pinching away. Here we clearly see the cytoplasm starting to move in, pinching away to form these two new cells. And we find that the nuclear membrane reappears here. We see the nuclear membrane reappearing, and we find, if we were to count the number of chromosomes in this, there's 1, 2, 3, 4. So we've got 4 chromosomes in that cell. And we've got 1, 2, 3, 4 in that as well. And at the end of cell division, we find at the end of telophase, when cytokinesis and karyokinesis is complete, we have 2 new cells, nuclear membrane developed, and these have the full set of chromosomes or nuclear material that the original cell had. So just to very quickly look at the characteristics of this, here we see a diagram showing you spindle fibers. Here we clearly see the formation of 2 new cells, and here you can see the nuclear membrane starting to reappear there, and that's obviously the concentration of the chromatin or the chromosomes. So cytokinesis starts by the cell membrane starting to constrict at the equator of the cell. So the cell membrane starts to constrict and starts to pinch away to eventually divide and form these 2 new cells. The nuclear membrane and nucleolus form. So we have the nuclear membrane forming around the chromosomes. Each daughter cell has the same number of chromosomes as the parent cell has. And that is essentially the end of the stages of cell division that we looked at. Now guys, very quickly we're going to go on to plant cells, and I'm going to show you a very good link to remembering this process. So we're going to explain plant cells very, very quickly. Again, interface. The only difference in a plant cell and an animal cell is that plant cells have a cell wall, and unlike an animal cell, you'll find that it will not be able to sort of pinch away. There's a process called invagination, cytoplasmic invagination. That does not happen because we've got a firm cell wall, and hence we find that when 2 new cells formed at the end of telophase, a cell plate will form. So I'm going to very quickly go through these slides with you with very little explanation, but I want to go on to what happens when cell division goes wrong. Here we're seeing a plant cell, fixed shape, cell wall, cell membrane, nucleus, chromatin network in the middle. So that's the cell in interphase. Again, we said that the DNA replicates and duplicates. Here we're looking at prophase, the first stage of this. Here we find the nuclear membrane disappearing. We find the formation of spindle fibers, and we find the formation of chromosomes, and these start arranging themselves and preparing themselves for the next phase, which is metaphase. So we look at metaphase again. Here we're seeing metaphase, chromosomes meeting along the equator in a single line, and here we clearly see this electromicrograph showing you the chromosomes along the equator here. Okay, as we go through the next stage after metaphase, after anaphase, sorry, go back, guys. We had anaphase here, and we see that here the chromatin material, or here we find that the chromosomes start splitting up into daughter chromatids from the equator and moving to the opposite poles. And this is where we find that it's slightly different now that we come to the end of telophase. Here you find the formation of the cell membrane, there's chromosomes in there, and here we find that because it is a plant cell, we do not have a cell membrane, and that the membrane can constrict, but we have what we call a cell plate forming. I'm going to show you that diagram now. Okay, sorry, very quickly, let me just go back there. Oops. Here we're seeing a cell plate will start forming, and essentially this is what happens. So here we're seeing, here's this plant cell, and here we find the cell plate, and that clearly can be seen here, and that's a new cell. So this is an electron micrograph, guys, of a plant cell at the end of cell division, and these will essentially split up at the end to form two new cells that are unconnected. So cytokinesis starts by a cell plate forming, which is a cell wall forming at the equator of the cell, which actually splits the cell into two halves. Each daughter cell has the same amount of chromosomes as the parent cell, and this will continue with the cycle of the cell. Okay, so what happens when cell division goes wrong? Because cell division is a constant process, and sometimes it does go wrong, and that is when we have problems and we have abnormalities that develop. And the term that we refer to when cell division goes wrong is cancer. It's a very prevalent topic, and we know that cancer affects lots of us, women, children, men, adults. No creed, no caste, no bar, no race, and it's something that it affects us all. We all know of people that are affected by cancer, and that's important for us to understand. And as Nock said, it's helping us. Life sciences helps us to understand what cancer is. And as a life science student, you'll be able to explain what is cancer to somebody who may not know, to one of your grandparents who may know that they probably have cancer but not understand the concept of what cancer is. So essentially, cancer is when a cell, each of the cells have a signal, or a signal that actually stops cell division at a specific point and then initiates it again. But however, when there's a cell division, when a cell undergoes between 20 to 50 times, it'll eventually stop dividing, and that cell is replaced by a new cell. Unfortunately, when it goes wrong, these cells do not stop dividing, and they constantly divide. And that constant division of cell leads to an abnormal mass of cells, which is not normal, and that becomes cancerous. So cancerous refers to cells that cannot stop dividing, uncontrolled cell division, and these, depending on the type of division and where it is found, we get different types of cancer. So cancer is dangerous because it can spread and attack healthy organs in the body. So very quickly, there are two terms that we refer to when we're looking at uncontrolled cell growth, and refer to them as, these will form tumors or clump of cells, and here we clearly see an accumulation of cells, guys. This is an abnormal growth of cells, and that can be removed because it is benign. Benign simply means that it is a tumor, it's uncontrolled growth of cells that have not spread to other parts of the body, and hence we refer to this non-cancerous or benign tumor. Here we're seeing a malignant tumor. And if you look at malignancy simply means the ability to spread. So here we're seeing that the cells are shed and they travel to the blood to infect other healthy tissue. And that is when the early diagnosis of tumors, I know women are encouraged to have regular examination, pap smears, self-examinations of their breast, to ensure that they pick up tumors early. And that's important. If you go to a clinic, if you go to a doctor, you know that they encourage this, and it's important. The thing is, you need to talk about this, talk to your parents, talk to your friends, guys, and this is it. You can, if it's treated early, if it's diagnosed early, it can be treated, and treatment can be very efficient and very successful. We all know of somebody that is affected or was affected by this. There are different kinds of cancers. Okay, carcinomas, sarcomas, and leukemias. So leukemia is basically, we've heard of leukemia, and that's basically cancer of the blood or the lymphatic and the immune system. Sarcomas are affecting bone and cartilage and the muscle, and your carcinomas are what affects the skin and epithelium. So if you have skin cancer, it's basically a carcinoma. There are various causes of cancer, but anything that causes cancer is called a carcinogen, and we refer to that as a carcinogenic substance. Very quickly to look at, so we could inherit cancer, we could have a predisposition. There's a likelihood that if somebody in your family has had it, there's a risk that you may have it. It's not a guarantee, but there's a risk. And hence it's good to check yourself and to go for treatments and for checkups constantly. Radiation. Radiation is it's X-rays, harmful X-rays, exposure to the sun, ultraviolet sun rays, all increase your risk to having cancer. Smoking, guys, is another factor that causes. It's not actually the process of smoking, but it's what is present in the contents, the chemical contents of the cigarette. Hormonal imbalances can also lead to that. We have pollutants in the environment that can lead, as well as viruses that also stimulate. Processed foods as well. We looked at lots of processed foods, guys. Eating too much of them can also make you susceptible to having cancer. Viruses, guys, I discussed that there's certain viruses that actually increase your chances of your papilloma virus, so HPV vaccines, and that's exactly a vaccine for a virus that actually increases your risk of having cervical cancer. So there are two types of tumors, guys. We discussed that. We said benign tumors that basically can be removed, that do not spread, and the chances of this being diagnosed early it goes well with treatment and early diagnosis. Malignant tumors are often cells that are cancerous and they leave the site where they initially started and they infect other healthy tissue. And that process of when it spreads is called metastasis. So you're going to come across these terms, guys, but you're going to be probably a few steps ahead of the other guys that are not watching. So invite them to watch along with you. And so there's different treatments, guys. And the most important, there are 3 different types of treatment when somebody does have cancer. You could surgically remove the tumor if it's diagnosed early. At a specific organ, if you can survive without that organ, yes, it can be done. So mastectomies are done, prostate examinations are done to remove cancer or tumors. Chemotherapy is done, so it's a combination of chemicals that are used, as well as radiation is done. So radiation basically is the injection of compounds and capsules with radioactive dyes in them that actually burn, which actually destroy. But each of these treatments have side effects, but those side effects are negligible to the outcome, sort of increasing your chances of survival and curing the disease. There's also traditional methods of treating them, such as herbs, as well as living positively, praying, being healthy, having a positive mind and focus about your health and condition will actually improve. Okay, guys, as we always end the session, we have lots of revision questions. I might not be able to go through all of them, but I know that there's many questions that I've designed for you here. I want you guys to go through, look at them, to attempt them, to try and answer them, be one step ahead. Here we're seeing a photomicrograph of cells, of various cells in different stages. And that's essentially what your teachers are going to put. They're going to put down photographs or electromicrographs of cells in different stages. You should be able to identify. So I'm going to very quickly probably answer one of these questions just to help you. So the question is, identify whether cell division in the above diagram is occurring in a plant cell or an animal cell, and give a reason for your answer. And if we look at this diagram, guys, we can clearly see here we're seeing a cell that has a fixed cell wall and a cell plate. So answer these questions, guys. It's the end of the session. Went fast, but lots of learning that has happened. I'm going to hand you over to Indy.
I can't believe that it's the end of the show, right,
I'm really
Knox?
sad, actually.
I know. Okay, but guys, grade 10s, thank you so much for tuning in. Grade 11s, don't forget that you are next. Thank you so much for such an amazing show. And Knox, I think we'll be seeing you in the grade 11 show too, right?
Yes, guys, make sure that you stay tuned after this, because you'll be seeing more of me on the grade 11 Life Sciences show.
Fantastic. Okay, guys, see you same time, same place next week. Goodbye.
Shop, shop.